Now, welcome to all of you for today's session of plant physiology and metabolism. In this lecture, we are going to discuss the deficiency and toxicity of zinc, molybdenum, and nickel. So, we will discuss one by one. First, what is the specific deficiency symptom of boron? You see that boron is a very essential micronutrient required for the proper plant growth and development, and it is a very unique element in the periodic table and having properties of both metal and non-metals. And boron is, like other essential mineral element, is involved in various structural and functional integrity of like cell wall and membranes, ion fluxes, cell division, elongation, and various metabolic processes of the plants. So, in this context, we will also discuss how boron is taken up by the plant. Generally, you see that sources of boron are basically organic matter and borax, and it is generally absorbed from soil as undissociated boric acid, that is S3BO3, and at pH less than 8. So, boron is basically taken up by the plants through three mechanisms, namely passive diffusion, facilitated transport by boron channels, and active transport. So, this boric acid is basically is a small uncharged molecule and is generally relatively permeable across the biological membranes. Therefore, the passive diffusion of boric acid is considered to satisfy the need of the plant requirement for boron, where boron is available in sufficient quantities. However, when availability of boric acid is limited, plant use boric acid channels for transport of boron. Now, what are the important roles imparted by boron like other essential mineral elements? Boron basically plays very important role in plant growth and development, and basically boron is required for synthesis of cell wall components, particularly pectins, and pectins are basically important for cell wall structure and function, affecting the expansion and growth. In addition to this, boron has been found in about localized, you say more than 98% of boron is localized in the cell walls of the leaf. And in addition to this, boron also facilitates the movement of sugar within the plant, which is very crucial for various metabolic processes and overall growth. Boron is also important for the process of reproduction in plants, particularly including pollen germination, pollen tube growth, and seed and fruit development. Basically, presence of boron is very critical, particularly for ensuring successful fertilization and production of seed. Boron also help in uptake and utilization of calcium by plant cells, and calcium is you see that important for cell integrity of the cell membrane and signaling process within the plants, and it is also involved in the activation of various enzymes involved in different metabolic pathways, and basically this calcium acts as a cofactor for various enzyme for proper functioning of these enzymes, where this boron acts as a cofactor. Now, in case of boron deficiency, plants reflects various deficiency symptoms, and you see that boron is basically in those regions with high levels of rainfall. Basically, boron is washed away from the soil in the form of BOS3, and therefore, the accessibility of boron to plants basically diminishes as soil pH increases, especially in calcareous soils and soils with high clay content. So, this decrease basically is likely due to the formation of boron hydroxide and subsequent absorption by anions. And deficiency of boron leads to various physiological diseases like rolling of leaves in potato and rotting of sugar beet, brown heart of turnip, cork formation in apples. So, you see that this is the tomato plant in the slide having deficiency of boron, and you see that there is a leaf rolled in upward direction, and there is a cup like formation appears. And if I talk about what are the specific changes induced in case of boron deficiency, it leads to various anatomical, physiological and biochemical changes, and these changes include inhibition of apical growth, necrosis of terminal buds, reduction of leaf expansion, and breaking of tissues due to brittleness and fragility, abortion of flower initials and shedding of fruits. So, very important anatomical deficiency symptom of boron have been associated with the cell wall abnormality and numerous physiological and biochemical effects that has been observed in case of boron deficiency, particularly like death of root and shoot tips, abscission of flowers. It also affects the nodulation in legumes, interveinal chlorosis, marginal necrosis, and infolding, like just you see in cup shape formation of the tomato leaves. It also affects the process of pollination and reduced internodal elongation. And due to reduced internodal elongation, plant generally attains compressed or rosette appearance. You see that, but the deficiency of boron is very much could be easily observed at the particularly terminal buds or youngest leaves, and which become deformed depending upon the type of plant species. Basically, boron deficiency could also lead to reduced fruit set, which is a well known symptom of boron deficiency. Now, what happens opposite to it once there is a toxicity of boron? Boron toxicity could lead to range of symptoms, and these symptoms you see in the slide, that leaf margins and tips which may progress to necrosis and premature drop of leaves. In some plants, boron accumulates in older foliage, leading to yellowing, browning or blackening of leaf margins or tips. And so, you see that boron toxicity is how much lethal to the plant. Now, next essential mineral element is zinc, and zinc is also one of the important micronutrient crucial for the growth and development of plants. And it is one of the second most common transition metal in living organisms after iron. And the average concentration of basically zinc in cultivated soil is about 65 milligram per kg, and zinc is found solely in the form of zinc second, and you see that in various metabolic functions zinc plays very important role, and you see that it is localized, it is distributed in the cytoplasm, nucleus and cell membrane, and can tightly attach to metalloproteins as a structural component, to metalloenzyme as a cofactor, and zinc could be compartmentalized into intercellular organelles and vesicles for storage, acting as a reserve for zinc dependent proteins. And in addition to this, zinc plays a very crucial role in various of course physiological processes in plants, including enzymes, protein synthesis, metabolism of carbohydrate and regulation of hormones, plant hormones. And basically plants require zinc in very small amount as compared to nitrogen, phosphorus and potassium, but its deficiency could result, or you can say could lead to uh, basically adverse impact on plant growth and yield of the plant. Now, how zinc is taken up by the plant? So, zinc exists in various uh, form as in the soil, like zinc oxide, zinc sulfate, zinc chelates, and primarily absorbed as divalent cations, and but it could also be absorbed as a monovalent cation. And basically plants absorb zinc from the soil through roots via facilitated diffusion, basically facilitated by membrane potential and transporters. Occasionally organic ligand zinc complexes are also employed for its uptake by the plant roots. But during long distance transport through xylem, zinc is either attached to the organic acid or present as a free divalent cation. In the phloem sap, zinc concentration are relatively high. Possibly it could be due to zinc being complexed by low molecular weight organic solutes. Now, what is the specific role imparted by zinc in plants? You see that zinc impart various specific roles in various physiological processes and serving as a very crucial structural, catalytic and signaling component in plant. As a essential plant nutrient, zinc serves as in part of various enzyme cofactor of more than 80 enzymatic enzymes, and these enzymes are involved in various metabolic processes. In addition to this, zinc you see that play a very important role in the formation of chlorophyll, and basically it prevents degradation of chlorophyll. And zinc is also a very key component of carbonic anhydrase, and basically it also regulates the conversion of carbohydrates and utilization of sugar. And in addition to this, zinc also contributes to the formation of tryptophan synthase, which is responsible for synthesis of tryptophan. And tryptophan, you see that you might be aware that it serves as a precursor to one of the important plant growth hormone, that is indole acid. So, any deficiency of zinc could affect the synthesis of indole acid. And as I mentioned that zinc activates numerous enzymes including dehydrogenases such as alcohol dehydrogenase and carboxylases. So, in protein synthesis, basically zinc is involved in the stabilization of ribosomes and forming a structural part of RNA polymerase. So, it is also associated with important enzyme superoxide dismutase, and in addition to this, zinc is crucial for maintaining the structure and function of DNA transcription factors including zinc finger, zinc cluster and ring finger domain. And during ripening and seed production, zinc basically promotes pollen formation and viability, and of course it is going to be translated into better yield of the plant in case of sufficient amount of zinc. Now, we will discuss some of the enzymes which contain zinc. Now, one important enzyme which is alcohol dehydrogenase, and basically this alcohol dehydrogenase enzyme is very peculiar in case that it contains one zinc atom per molecule. Sorry. This most of the enzymes generally contain only one zinc per atom, but alcohol dehydrogenase is an exception which contains two zinc molecule per atom, two zinc atom per molecule. Out of these two zinc atoms, one atom serves as a catalytic role, while the other fulfills the structural function. The catalytic zinc sites are coordinated by two cysteines, one histidine and one water molecule, whereas the structural zinc binding sites typically involve four cysteines. So, alcohol dehydrogenase basically facilitate the reaction, that is conversion of ethanol to acetaldehyde, which is then further oxidized to acetate by aldehyde dehydrogenase in subsequent reaction. Then another enzyme you see, that is carbonic anhydrase, which is also a zinc containing enzyme, plays a very crucial role in catalyzing the hydration of carbon dioxide. What happens in dicotyledonous plants, carbonic anhydrase comprises 6 subunits with a molecular weight of about 180 kilodalton, and each molecule contains 6 zinc atoms. So this enzyme carbonic anhydrase is basically found in both chloroplast and in the cytoplasm. And now what question arises, what is the role imparted by this carbonic anhydrase? So basically this carbonic anhydrase has very important role in chloroplast, particularly in C3 and C4 plant, and basically in C3 plants this carbonic anhydrase basically facilitate the diffusion of carbon dioxide to the sites of carboxylation by Rubisco. But in C4 plants, carbonic anhydrase basically facilitate the conversion of carbon dioxide to bicarbonate, which is subsequently utilized by phosphoenol pyruvate carboxylase. So this enzyme carbonic anhydrase plays a very pivotal role in unique carbon fixation pathway of C4 plants, which help in efficient assimilation of carbon dioxide. In addition to this, zinc also serves as a metal component in several enzymes, for example, alkaline phosphatase, phospholipase, and these two enzymes basically contain 3 zinc atoms, each of which at least one has a catalytic function. And then carboxypeptidase, which hydrolyzes peptide cleavages and contains also single zinc atom which with catalytic functions. RNA polymerase, which contains 2 zinc atom per molecule, is inactive if zinc is removed. Now, what are the specific deficiency symptom of zinc? Basically, you see in the slide that zinc deficiency in dicotyledonous plant is generally very specific, and generally deficiency symptom leads to stunted growth as a result of shortening of internodes, leading to a very important phenomenon known as rosetting due to shortening of internodes. Generally, zinc deficiency could also lead to reduction in leaf size, often termed as little leaf, and observed in many plants. Severe deficiency of zinc can lead to death of shoot apices, and this is referred to as dieback disease. And this dieback disease has been observed in forest plantation in South Australia, and generally zinc deficiency leads accompanied by chlorosis, and very important deficiency symptom mottle leaf disease has been observed. And you see that zinc deficiency basically impact also grain and seed yield to a greater extent than the dry matter production, and basically this reduced yield could be due to the impaired pollen fertility in plants induced by zinc deficiency. Generally, zinc deficiency could also lead to root exudation of low molecular weight solutes, and in dicotyledonous plants, amino acids, sugars, phenolics, potassium basically dominates, whereas in monocot plants, main solutes are phytosiderophores. So these are the specific deficiency symptom induced by zinc. Now, what are the toxicity symptoms observed in plants? Basically, zinc toxicity is very rare in crop plants because in soils contaminated generally by mining, smelting, or sewage sludge treatments are basically, and if plants are grown on such soil basically leads to the toxicity symptoms. So, high level of zinc supply can induce toxicity in generally non-tolerant plants with root elongation, with inhibition of root elongation, and which is one of the very sensitive parameter to measure the zinc toxicity. And zinc toxicity, you see that in the figure, that leads to chlorosis in young leaves due to the induced deficiency of other nutrients like magnesium and iron. So, toxicity basically induces deficiency of other essential mineral elements, like zinc toxicity induces deficiency of magnesium and iron. So, zinc toxicity also affects the process of photosynthesis, leading to impaired activity of particularly PS2 activity. Now, molybdenum is another essential mineral trace element and needed in very small amount. So, generally molybdenum generally binds with pyrenoprotein and could lead to the formation of molybdenum cofactor. So, this molybdenum cofactor plays a very crucial role in sustaining the activity of more than 60 enzymes. So, how you see that molybdenum is important for plant, but it is required in a small quantity. So, generally 50 grams of molybdenum per hectare is sufficient to meet the requirement of a risk crop. And generally this molybdenum is found in farmyard manure, seeds or other planting materials, as well as could be present in the trace amount in certain artificial fertilizers. So, now how question arises, how molybdenum is taken up by the plants. So, basically this plant make availability of molybdenum in the predominantly in anionic form of molybdate, and generally this is released into the soil from solid minerals through regular withering process and then undergoes various reactions. So, solubility of molybdate is significantly influenced by soil pH. It is one of the very important crucial factor. So, generally in acidic soil you see that molybdenum tends to be inaccessible to plants, leading to higher prevalence of molybdenum deficiency compared to neutral or alkaline soil. And generally it is very interesting to note that molybdenum exhibits increased plant availability as soil pH increases, and the solubility of molybdate increases approximately 100-fold for every unit increase in soil pH. Hence, the application of you see that lime is very important to raise the pH of the acidic soil, because it is one of the strategy to enhance the availability of molybdenum. Molybdenum uptake increases with liming of the soil. Addition of lime increases the soil pH. So, generally applying phosphate also helps to release the adsorbed molybdate from iron oxide due to phosphate's strong affinity for molybdenum, and thereby boosting the water soluble concentration of molybdate in the soil. So, you see that molybdenum is also necessary for maintenance of proper activity of more than 60 enzymes, and generally crystallographic analysis of molybdenum enzymes have revealed that the molybdenum cofactor basically resides deeply within the holoenzyme structure. You see in the slide, molybdenum serves as a vital component of nitrate reductase enzyme, which converts nitrate ions to nitrite ion, and molybdenum also plays a role in purine breakdown and acts as an integral part of the oxidase that transform abscisic acid aldehyde into abscisic acid. In addition to this, molybdenum contributes to the sulfur metabolism by facilitating the oxidation of sulfide to sulfate. Then you see that nitrogenase is a very primary enzyme complex responsible for nitrogen fixation in all you see nitrogen fixing microorganisms which contains molybdenum nitrogenase. The nitrogenase reaction basically necessitates energy in the form of ATP and reducing equivalents or flavonoxins. So you see that in the slide, the sequence of reaction catalyzed by molybdenum nitrogenase have been shown in the slide. That another enzyme which requires the presence of this molybdenum is nitrate reductase, which is a homodimeric enzyme and have possess 3 electron transferring prosthetic group per subunit, namely flavin, heme and molybdenum cofactor. And molybdenum cofactor consists of 2 molybdenum covalently bound to 2 sulfur atoms in the tricyclic molecule pterin, and molybdenum in molybdenum cofactor is bound to a third sulfur ligand either of cysteine residue or of terminal sulfur. So you see that molybdenum cofactor is the first type used in nitrate reductase and sulfide oxidase, and the second type of is found in xanthine dehydrogenase and aldehyde oxidase. And during nitrate reduction, electrons are transferred directly from molybdenum to nitrate. And in addition to this, there are 3 additional molybdenum containing enzymes that have been discovered in plant, namely xanthine dehydrogenase, aldehyde oxidase and sulfide oxidase. These enzymes are crucial for plant response and resilience to diverse stresses. Now, what are the specific deficiency symptoms of molybdenum? And this specific deficiency symptoms in non-leguminous plants include stunted growth, failure of leaves to develop to healthy state, and pale green or yellowish green color between veins and along the vein edges appear in the leaves, you see in the slide. And generally advanced stage of deficiency lead to dead leaf tissues at the margins, particularly affecting the older leaves. So in cauliflower actually exhibits yellowing of outer leaf tissues followed by death of hard leaves edges, resulting in the formation of narrow distorted leaves, but very specifically known as whiptail disease. So generally the deficiency could lead to intravenoidal marginal leaf chlorosis, and death of tissues are generally observed, particularly scald in French beans have been observed. And generally main symptoms relate to molybdenum deficiency in legumes, particularly its inability to fix atmospheric nitrogen, which could lead to stunted growth and yellowing similar to nitrogen deficiency. So you see that how molybdenum is important for plant. Now nickel is one of the trace element, and generally it comprises about 0.008% of the earth crust and present in the soil, and this is present in the soil and required by the plant in the process of biological nitrogen fixation. And therefore any sort of deficiency of nickel going to affect the plant growth and metabolism in many ways. Generally nickel is required by the plant in very minute concentration for normal growth, but high concentration may exert deleterious effect on plant growth and produce symptoms of toxicity. So now how nickel is taken up by the plant roots? It could be through both passive diffusion and active transport mechanism. However, you see that the proportion of uptake, nickel uptake through these mechanisms varies widely depending upon various factors like the plant species, the chemical form of nickel present in the soil, and the concentration of nickel in the soil or nutrient solution. For example, soluble nickel compounds are predominantly absorbed passively using cation transport system, and in contrast to this, chelated nickel compounds are basically absorbed through secondary active transport method, as certain transport proteins like permeases have a specific affinity for binding to nickel. Opposite to it, insoluble nickel compounds enter plant cell mainly via endocytosis. In biological system, generally nickel is primarily found in its oxidation state, that is nickel 2, and it can exist in various redox state like in nickel 1 and nickel 3rd, but it is absorbed by plants in the form of nickel dicalen nickel 2 ions. Now, there are certain enzymes where nickel is imparting a very important role, and these nickel containing protein is particularly metal coenzymes like you see that methyl coenzyme M reductase, superoxide dismutase, and nickel dependent glyoxidase and dioxinase, and carbon monoxide dehydrogenase, acetyl coenzyme A, decarbonylase synthase, and methylene urease. So, among all these enzymes, urease and nickel urease acid protein are particularly important for plants. Urease, which is generally isolated from jack bean plants, has a larger size and made up of 6 subunits, and each subunit of urease contains 2 nickel atoms, and in these subunits nickel is coordinated to nickel and oxygen ligands, and you see that how nickel is imparting a particular role in this enzyme urease. So, in case of nickel deficiency, it is going to affect the proper activity of urease enzyme. Now, what are the specific deficiency symptom of nickel? You see that nickel deficiency could affect the accumulation of urea, and it could lead to necrotic patches, and nickel deficiency also lead to decreased activity of urease enzyme, and of course subsequently leading to urea toxicity, and generally it is manifested in the necrosis of the leaflets. And generally nickel deficiency could also result in the marked enhancement in plant senescence and a reduction in iron concentration in the tissue. And you see that in the slide that how nickel deficiency results in deformed leaves, and particularly one specific deficiency symptom observed by nickel in case of nickel is known as mouse ear disease. So, this is all about the important role played by nickel in plants. So, we have finished mineral nutrition of plants and the specific role played by various mineral elements. Thank you very much for going into the details, and hope you will enjoy while going through my lectures. Thank you very much.